titled Genetics of Interconnected Populations of Small and Large Size, so pretty perfect for this meeting. She then went on to Melbourne, Australia for a postdoc before returning to Switzerland to become a professor at the University of Neuchâtel, then on to the University of Basel, where her lab is currently based. Dr. Willi has published extensively on research that explores the evolutionary consequences of small population size, the causes of range limits, and the evolutionary implications of plant mating systems. What we particularly admire about her work is how integrative it is. She draws on tools from evolutionary genomics and experimental ecology to understand the impacts of population size on adaptive and deleterious genetic variation. And that's sort of a theme we had from yesterday, is really wanting to connect genotypes to phenotypes. And so it's really exciting to hear about that. So we are absolutely thrilled that she can join us to share her insights on how population bottlenecks impact mutational load and adaptive potential in Arabidopsis. So please join me in welcoming Dr. Willi.
Thank you very much. Maybe, do I need a microphone? Yes. Carry it around.
Or you could try to your lanyard to this.
So, do you hear me? Is it okay? Noise? Perfect. Yes, sorry for not being ready this morning.
Excuse me. Answer.
Yeah. Okay, thank you. So, thank you very much. Thank you very much also to the organizers for bringing us all together. It has been a super exciting day yesterday, and I'm very much looking forward to what's coming. Today I will talk about something, and I will already give you the sort of get-away message, and I hope I get this across. I do it right now. I think when we have populations of small size, or a history of small size, then often we have also these other implications seen on the entire genome that is basically then can lead to a decay in the phenotype, so negative consequences for the phenotype, and I would call this the drift syndrome, and this is basically the word in my title, or the term. We were looking for the drift syndrome. I think we are all looking on it, as we've seen yesterday from these talks. So my talk is structured in the following way. I would like to talk about the extinction vortex. We've seen it yesterday on the slide, the classic model evidence for it, then refinements of aspects of the extinction vortex, and again, I think many of us are working on this. I would like to draw then a synthesis of current research, talk a bit more about this drift syndrome, and then tell you what I think, in what way we should extend the extinction vortex, or extend the vortex model for conservation biology. But let's start with the vortex model. It was actually described first by, among others, the father of conservation biology, Michael Soule, in his book, together with Michael Gilpin, and they described 4 aspects of the vortex. It's a book about minimal viable population sizes. And basically we have as a starting point a small population, so often it's declining actually, because he had this conservation perspective, a decline in population size. This leads to more inbreeding, exposure to random changes in allele frequency, genetic drift, a loss in genetic variation. This is sort of the first important aspect that I will pick up later again. Then inbreeding depression, loss of genetic variation may lead to a reduction in individual performance, maybe population mean performance. So performance right now in the population, but then also performance in the close future, if there is environmental change, these populations should be less able to adapt. That may feed back in so constrained vital rates, reduced reproductive output, reduced survival, and then smaller population, and that's the sort of the Smirnoff effect. So there is these other two aspects: link genetic variation with population mean fitness, or here individual fitness written, and ability to adapt. What's the empirical evidence for that? And I have to say this right away. We've seen yesterday talks with lots of publications from very recently. Actually, these synthesis articles, none of them is particularly new. And the one on plants that I will briefly introduce you to was written in 2006. So it's only studies on plants. First, on this aspect of the vortex of synthesize genetic marker variation. What's the correlation? We expect a positive correlation, and across studies, so again, this is a metadata study by Limo et al. Overall, we see these correlation coefficients being in the positive range here. There is variability. There is variability among type of plant species, outcrossing plants, self-fertilizing plants. About plants, the following has to be said. Most of them are hermaphroditic. They could self, but they do not self-fertilize, but are obligate outcrossers. So these are the signs here: diamonds, outcrossing plants. The highest correlation coefficients we see here for outcrossing plants, and in regards to genetic marker variation, what was looked at is number of alleles and fraction of polymorphic loci. They seem to be aspects of the genetic composition of populations that seem maybe to respond earlier to a decline in or to variable population sizes in comparison to heterozygosity. Seems like, based on these results. Then a second aspect, low population mean performance in populations with low genetic diversity. So again, we look at correlation coefficients between genetic variation and now performance, population mean performance, and you see the number of studies that found overall positive correlation coefficients for different type of plant species again, or what was looked at, whether it was DNA-based marker variation, or at the time, because this was published in 2006, they relied also on allozyme studies. So here we see a bit less of an effect. Correlation coefficients is higher for DNA-based studies. And then the third aspect, this link between either synthesize or genetic variation and the ability to adapt. And this is another, this is now a real meta-analysis, a formal meta-analysis, again by Limo and Fisher. And what they did is the following. Again, only plants. And they looked at the signature found in populations, the signature of local adaptation. Now, how do we test for local adaptation? The classic type of experiment is a reciprocal transplant experiment. So you take organisms from many sites, for example plants, and then you experimentally plant them in their home environment, but also in away sites. And it's this effect of difference in performance between home and away sites that gives you an indication of the magnitude of local adaptation. And so they had this magnitude of local adaptation split into two categories found for either small or large populations. So I think for simplicity and to do the meta-analysis, it's easier to work with categories, small versus large. And you see these small populations, their signature of local adaptation is basically overall 0, whereas large populations tend to be locally adapted. So yes, there seems to be this link between now in this case it's synthesize and the magnitude of adaptation. overall, there is good support for these vortex models, the vortex model or aspects of it. There are many meta-level analyses out. Unfortunately, they're all relatively specific in the taxonomic group they're focusing on. So this on plants was relatively extended in its scope in this regard. Most meta-analyses are old, from a pre-genomics era, and now having seen all the exciting results that have come out recently, I think there is really an opportunity for now getting on board here and doing this meta-analysis on the different aspects of the vortex. I think that would be super important. That would maybe also be very strong for the field of conservation genetics to some kind of keep the stand that we have in science, but also in our how the outside looks at us as a field, what we're contributing. Okay, that's the first part. Then now about the second part. The vortex model was maybe a bit rough in terms of the connections it predicted, and we have this refined hypothesis that all of us who work in population genetics actually work with. And these are that small population size or increased genetic drift lowers genetic variation. So we have this decay of heterozygosity from generation to generation that goes at the rate of 1/2NE. But we have also, we have this loss of rare alleles or fixation of alleles that are at high frequency. That's sort of the implications for genetic variation. We have a reduced efficacy of purifying selection, less efficacy of negative selection, and therefore accumulation of deleterious mutations, mutational load. But obviously the same should be the case for positive selection, that the efficacy of directional selection is reduced, and this is sort of a cutoff relationship depending on the selection coefficients acting on alleles. So there's this threshold. If the selection coefficient is smaller than about 1/2NE or 4NE, there is this increasing effect of drift. And finally, we expect drift to lead to less independent segregation of loci, more linkage. So these are the specific hypotheses that we also tested, but the system is not a furry animal, or we work with plants. Actually, we work particularly with one species of plant. It's the rock or sand cress, and it's an Arabidopsis species. It produces basal rosettes that then produce inflorescences, bigger flowers than Arabidopsis thaliana. That's a selfer. This is now the outcrossing relative, pollinated, for example, by syrphids. And this is the distribution. It's a North American species. It occurs along the Appalachians here. So all the gray dots are localities reported since 1980. And then we see it occurring in the Midwest, in the Ozarks, in the South, and then Great Lakes region in the north. That's the distribution. Then the habitat. So I said sand, rock cress. It all depends on where you find the species. You either find it on sand, then it's the sand cress, or you find it on rock, it's the rock cress. It likes disturbance. So you see that, but it only occurs in relatively pristine habitat because it likes this intermediate disturbance, not too much disturbance as along roadsides, but sand dunes where you have regular build-up of dunes and erosion and build-up and erosion. Or here it's usually, you actually see the species here. It's not close to the water where there is regular wave wash. It occurs where there is rarely, when there is a dramatic storm, the waves come up, wash out this rock, and that's what the species likes. The species would also like to have wildfire, but because of wildfire suppression, it has declined across its entire range. It's actually, I find this exciting to see how wildfire used to be very common in this part of the country. So we see a lot of blue here. Blue here means understory fire, so these are not very hot fires. These are fires that go through the landscape extremely rapidly if they occur at the periodicity of less than 1 per year. No, more than 1, so 0 to 10 years periodicity, or to 34 years. So very regular fires. There is an entire element of the North American flora that is fire dependent, and most of the species do very badly, especially, I think, in the east. So about addressing now this hypothesis. So we start with small population size, drift, lowest genetic variation in our system, and we really tried to go for explaining all the variation in genetic diversity across this species range. Obviously, we didn't do perfectly well, but we did pretty well. So we started actually some time ago. As you see, the publication is not relatively new, and yeah, we started small. We started with northern populations that we selected, and we looked at microsatellite marker diversity. We had 19 at the time. We calculated expected heterozygosity, and we looked what are the local factors that are correlated with this measure of genetic diversity. And we found that actually we can explain 76% of this in just the outcrossing populations. You will later see that there are also selfing populations. And the important variables are census size, first of all. So here it's not so impressive. You see census size on the x-axis. On the y-axis, residual expected heterozygosity is a positive correlation, a lot of variation. What is stronger is the association between small-scale spatial structure and diversity found in these populations. The stronger there is the substructure is. So if you have relatively closely related individuals sitting together, and then there is a decay in relatedness, but there is the next peak of related individuals, etc., etc. The more you have such structure, the more expected heterozygosity there is in these populations. And then rock populations harbor more genetic diversity than sand populations. That's probably because they're a bit more stable. I have to also say, I use this term population now all the time. Yesterday I was taught one has to define it, and our definition, we have sort of a work definition of what a population is in the field. It's plants occurring relatively continuously until we have gaps of about 50, or actually usually it's 100 meters or more, and we call this our population. And usually the next population, it's not just 100 meters away, it's usually many kilometers away. These populations are nowadays extremely isolated. They're actually genetically also highly divergent from each other, even over short distances of a couple of 100 meters. Okay, then obviously we couldn't explain 24% of the variation. We thought, that has probably something to do with history, and we should actually sample the entire species range and see if we understand the history of the species range, whether we can explain more of this genetic diversity or learn more about the history of genetic drift in this system. So we sampled 52 populations from across the range. We did pool sequencing. We always took 25 individuals. It was Illumina parent sequencing. We produced a relatedness tree to understand the history of the populations and linked this with genomic diversity. So it was Tajima's pi in non-coding regions. And now first to the species history. We learned quite a bit from this relatedness tree. So you here see on the left a trimix tree, and it's very small, but basically what you see is an early clade with only 2 populations. These are the Missouri populations in green. Then we have 2 other colors here, blue and purple. Blue, the populations in the east, and here the other populations in the Midwest. Then what you see on the map here is the this dashed blue line, it's the maximum extent of the ice sheet during the last glacial max. And when you look where all these populations are, these gray symbols, most of them are north of this line. So there must have been a history of recent colonization. And then we looked actually, which is the last common ancestor of the first, or the common ancestor, just one note before the first population emerges in this tree that is nowadays in an area that was covered by ice. So do you see it? So, and basically we plotted this tree, or the ends of this tree then on the map, and you see that this common ancestor, when we plot the tree on the map, is located here. Quite nice, close to the dashed line. Here the same. So it seems like this species survived the Ice Age, or at least some of populations, others are older, close to the edge of the ice, and when the ice withdrew, it expanded into these northern areas towards the north, and then mainly eastward. For the western cluster and the eastern cluster, we have more a star-like expansion, including towards the south. So these southern populations are new, or no, young. No, not young. What else do we see on this? But basically, this suggests us this colonization route. We can also basically, along this tree from each endpoint, the population say what the distance of expansion was, if we take the projected tree here on the map. That's one thing. Then we see these circles, right? These are the selfing populations. There are also two mixed mating populations. They're a bit spatially aggregated. Here, for example, around Lake Erie. What we also see is there has been past gene flow, for example, in this Lake Erie region between the two clusters here, east-west, and here from the Missouri populations a bit northward to northern Missouri and Iowa. Okay, so I've explained everything. That's the history of the species, the recent history of the species. And then we look, okay, does this explain genetic variation, this history? It should, based on theory. We have in the southwest rear edge isolation, so probably these populations in Missouri in Iowa have been isolated for a while, so they should have more of a signature of genetic drift, less genetic variation. And for the northern populations, we expect maybe serial bottlenecks of during colonization having led to a decline in genomic diversity. So about now the next slide actually goes directly to the results. Yes, this is now what we found for Tachima's pie, the distribution across the range. And we see interpolated values on the map on the left, and this for outcrossing populations only. The selfing populations make just everything more extreme, and therefore we exclude them. So you see darker color means higher Tachima's pie, more genomic diversity. When you look at the two clusters, now we pooled these Missouri populations with, put it in the western cluster to make it simpler. You see we have dark in the center, and then this decline towards the edges. So this range edges, they have the signature of reduced genetic diversity, higher signature of genetic drift, a past of drift. And actually, when you use this distance to core that I explained you just before, distance to core, it can be either rear edge isolation, so it's got just the great circle distance from these southern populations to the triangle here, or it's from the endpoints of colonization back along this trajectory to the triangle. This distance to core is negatively correlated with Tachima's pie, and it's actually quite a strong correlation. So yes, this history explains a lot of genetic variation, at least for Tachima's pie. Then what about mutational load? So in these populations with a history of drift that is pronounced at range edges, I have to say also there is an exception. Maybe here in the east, this decline in diversity is stronger towards the south, but here in the north, for example, that didn't work. But it's probably just these expansion distances were too short. Okay, about mutational load. So we wanted to do the same as we did for Tajima's pi, but for mutational load. Now we used several estimates of mutational load based on SIFT algorithms, SNP-F. I only show you maybe the least sophisticated estimate, but it's actually a good one. And it is, so we look at exons only, we carefully filtered for low frequency SNPs, and then use this estimate. So it's the number of polymorphic non-synonymous to synonymous sites, so only polymorphic sites, assuming non-synonymous ones are probably often deleterious. Plus, we adjusted this ratio by the ratio of mean derived frequencies. So we use an outgroup, Arabidopsis thaliana, so we know what is the sort of ancestral state, what is the derived SNP version, and we take of the derived SNP version the mean frequency, and that ratio is then used for adjustment, and it gives us very, very strong results. So we see the same two type of plots again. On the left you see dark gray means a higher estimate of mutational load. We see gray color at range edges, and in the east here, especially towards the south, but not very much towards the north. And actually, when we take these outcrossing populations again only and plot this against distance to core, there is this positive relationship. So the longer the expansion history, or the longer this rear edge isolation from these cores, the more genomic load. Then we looked at positive selection, and we used a relatively crude, but I think not such a bad estimate as well, of positive selection. Well, you have to decide whether it's good or bad, actually, not me. But it's alpha. So it's based on the McDonald-Craigman test. And the McDonald-Craigman test does the following. So you look at coding regions only, and you look at the fraction of non-synonymous substitutions. So again, you need some outgroup. In our case, it's always Arabidopsis thaliana as the reference. Non-synonymous substitutions, many of those should have been fixed by positive selection, and this relative to substitutions synonymous. And as the other reference is the same ratio, but for polymorphic sites. So non-synonymous polymorphic sites to synonymous polymorphic sites. Now you would think, oh, we are in a session of small populations, and we know the small populations drift should increase the polymorphic non-synonymous sites because purifying selection being less efficient. And Messer and Petrov came up with a very clever idea of getting around this problem, that the reference is not ideal anymore. And basically what you do is you take the derived allele frequency, so you take site frequency spectra, unfold it, and you look for different classes of polymorphic sites. So you take these polymorphic sites, calculate the reference, and you look at the behavior of alpha, which is derived from this McDonald-Craigman test. You look at the behavior of alpha, this rate of substitution, over the frequency spectrum. And you do expect, yes, here we have bad reference of the polymorphic sites, but it should get better and better, and at the asymptote you probably do well. And that's where you estimate, how you estimate alpha is at the asymptote. Okay, that's what we did. And here again you see how alpha is distributed over the species range. This signature of positive selection, dark means high signature, dark tends to be in the center, also in the east, but again the pattern is stronger for the south compared to the north. And we see with distance from core, the signature of positive selection declines. So at the range edges, and in these populations with a history of genetic drift, we do not have only a problem with mutational load, genomic load, we also have a problem that these populations seem to be less well adapted, or they seem have less of a signature of adaptation. Then we did the same thing for LD. Here we looked at R square, so correlation among variable SNP positions. We only had short fragments, but we did it for the paired ends and fragments. And you see that, so high correlation coefficients means high LD. It's darker, higher values tends to be the case for range edges as compared to center. And again, here the north has high low linkage disequilibrium, the south higher. So to sum up about these refinements, yes, when we have populations with a history of genetic drift, and again this is over an entire species range, and this is a drift that probably happened during expansion, so many thousand generations ago, we see this imprint. We see this imprint in genetic variation, Tajima's bi, but we still see this imprint in higher genomic load, lower directional selection, evidence for directional selection, and higher linkage. And that brings me to this idea of this drift syndrome. It would be nice, and yesterday we heard Tajima's D may also be such a good estimate that captures the history of drift, maybe more recent drift. And its association, or I also saw yesterday a poster about rattlesnake trying to do the same. Sort of, you have a genomic estimate of drift, genetic diversity being reduced, and what is the correlation between these other aspects also affected by drift? And actually what we find is these correlation coefficients are extremely high. So for load, this estimate of load, it's minus 0.92, so I just now say it positively, 0.5 for alpha, the signature of directional selection, and similar size correlation coefficient for LD. So very strong correlations. And I think with genomic data, we're going towards that. What I showed in the beginning, these plant studies, obviously once you go to phenotype, it's a different story, but as long as you stay within genomic data, I think these correlation coefficients are probably very high. In our case, they are. But then, did I have a sorry? Yes. So obviously this is now orthogenomics, and it's sort of, you know, when you do so, and I think this is also what we have a bit of a problem if we work in conservation genetics, to bring this part of our knowledge out to managers, because they think, oh, this is genetics, you know, what does it tell us about the phenotype? And I think, yes, this link to the phenotype, to expression is very important, but it's also super hard, because we may have the idea, oh, it would be nicest to show really causally, here is the mutation, here is the phenotype that suffers. And I think for load especially, this will probably be never possible, is my feeling, because we talk of this accumulation of slightly deleterious mutations. To detect the effect of slightly is minimal. So probably we have to go just for a rough approach and stay on the level of populations. And that's exactly what Dario Sanchez did, and Antoine Perrier, he's now a postdoc in Virginia. They basically used these populations, some of it, they selected 20, and these 20 were selected based on the amount of genetic variation they had and estimates of mutational load, and then they looked phenotypically how well they do, and they do it in a very specific manner. So they took plants of these 20 populations and performed within across these among plants within the same populations, but also with plants from different populations, so between population crosses. And the partner populations for West, it was actually this, and for the East, it was this one. For Eastern populations, it was this population. So they always used the same sort of crosses to the same background, a different one for East and West. And what you can then do is you raise this offspring, and when you find a difference in performance, it's what we call the heterosis effect, it should be, at least to great extent, due to the masking of deleterious recessive mutations. So yes, they're masked because they then occur in a heterozygous state. That's the heterosis effect. Between population cross performance minus within and relative to within. And they did raise this offspring, these were thousands of pots and plants at 5 sites throughout the range. They started with having the plants under the tent. They removed the tent. It was just for sowing and initial germination because we wanted to have a good start. We didn't want to have the effect of a catastrophe right at the beginning. Catastrophe means heavy rain, for example. Here you see the result. So heterosis was based on an estimate of multiplicative performance, which was germination rate times reproductive output estimated over 2 years in this garden experiment. And each dot here is a population plants estimated at one site, one common garden, so the 5 colors. And here is a heterosis in this multiplicative performance. So it's +1 to bring it up, so that means basically 0 heterosis effect. That's the baseline, and you see this positive relationship. It doesn't look so impressive as it is in reality, because here actually we work with a log transformed scale. So this heterosis effect is actually huge. And when you look just at the within population crosses, without taking this reference of between, this within population crossed plants of populations that come from range edges, they have up to 80% reduced performance. So really significant. So what about evidence for reduced local adaptation? So we didn't quite have the design, or they didn't have quite the design of home away, as I mentioned before, reciprocal transplant experiment, but they had these plants being raised at five gardens. These 5 gardens differed in conditions, for example in temperature. Some of these temperatures were then closer to what the plants experienced at their home site. So here you see on the x-axis the difference in temperature between now I have to think, so it's the home site minus garden conditions, I think. Now you will see, now it's the other way around. Anyway, it is, so 0 means it's like at home. Minus means it's cooler in the garden than it was for these ancestors of the plants at home, and here it's warmer. And this for plants varying in populations that vary in Tachima's pi. So it's a sort of a 3D plot, not quite. So we see a contour plot. We see multiplicative performance. Red means very high performance. We see that all plants do extremely well when it's cooler in the garden than at the site, at their home site. And it basically means it's a cold adapted plant, and it occurs now in sort of under marginal conditions, which is probably not uncommon. Interesting is what happens at 0. So we see here not much of a difference across this gradient of genetic diversity. Populations do about equally well. Maybe if you want, it's a bit more reddish here, and here maybe more bluish, which would be an indication of reduced signature of local adaptation in these populations of with a history of genetic drift. But overall it's significant, but only when we test across the entire gradient of delta temperature that these small populations with a history of small size, they tend tend to do less well, and what is especially obvious here, that if it gets warmer, they do less well than populations with a history of large size. And we looked at other maybe spurious correlations with the conditions from which these populations come, and it seems to be robust. So this reduced adaptation in long-term small populations, there is not so strong evidence for this, or especially in comparison to what I showed before on genomic load. So if we come back to this idea of the drift syndrome, then probably with this alpha, we don't know very well, probably it doesn't have strong phenotypic implications, this reduced signature of positive selection. What has certainly implications is this increased signature of mutational load, because here this is from another experiment where we showed this, actually this increased load translates into reduced lambda. So when we do a basically do life table study and estimate population growth rate, we see that with increasing genomic load, we have reduced population growth rates. So it does have a very strong phenotypic effect. It's very hard to work with sort of to translate this in maybe vital rates, that's still possible, but then the vital rates into population growth rate, you have to make assumptions. And so therefore I actually like to show just the results on multiplicative performance. Yes, so to sum this up, once populations start losing genetic variation at the level of heterozygosity, they have also been affected by increased mutational load, decreased positive selection, heightened linkage. We have these associated performance declines, measurable but stronger for load than for reduced adaptation. Yes, so now about this extension of the vortex model. So I think the vortex model, so this is from the textbook of Primac representation of the model. I think here it would be fair to make more specific predictions, and actually we all work already on these very specific predictions. We shouldn't just say, oh, we have more genetic drift, less ability to adapt. This is such a vague, it's a relatively vague way of describing the processes that are expected to be ongoing. So we should be specific and say more mutation load, less adaptation, higher genome-wide linkage, and maybe other estimates that you want to put forward. What I think, and Christina introduced this yesterday very nicely when she said NE is not NE. There are basically NE depending on how you estimate it. It refers to different times back in the past. I think when we go out and we estimate, so it's very hard to estimate census size, and census size is often not so strongly correlated with genetic diversity, because the history of this species, the history of range dynamics, for example, they have such a strong imprint on the species-wide distribution of genetic diversity that we have to keep this in mind. We have to know where we are in time and in space in regard to species history and its distribution. I think that is an important thing. Maybe as a nutshell, just that I think that we're all aware of this again. I think this is an amazing insight that you have something that happened 20,000 years ago. For this plant, this maybe 10,000 generations ago, and we see this imprint of genomic drift. It is has phenotypic implications. We still see this. Obviously, these populations can do well despite not being on the super fitness peak. So probably a lot of what is living out in nature is not on its absolute fitness peak that it could have, as that we can also see in this species. And then I think an important point I want to make is, so it's said we live in a time of extinction crisis, a biodiversity crisis. What we see already is local extinctions, maybe more rarely that entire species go extinct. we live in a phase of extinction crisis, but it's not only on the level of populations or species. What we also see is that we have a decline in positive species interactions. And okay, plants have their pollinators, or they have below ground mycorrhizal interactions, so positive interactions are important, but positive interactions are important for all species. We live as consumers for something that lived in the trophic cascade below us. So we have the unilaterally positive interactions, the bilaterally positive interactions. We have in ecology a lot of positive indirect effects, for example, over trophic cascades. So this loss of positive interactions, I think, should be included in this vortex model, and it can just be, for example, by when we see there are close associations. The plant that depends on particular guild, maybe, of pollinators. What's going on with the pollinators? So we should look at the co-spiraling of species, especially those with which positive interactions are shared. And with this, at the very end of my talk, yes, the extinction vortex, I think the empirical support for it would need an update, and that would be terrific for the field, I think. We all work now on this refined model, and when we apply it, and maybe that's something also that is coming out now, that we see these strong correlations across these aspects of this, call it drift syndrome, call it something else, and it has phenotypic implications, yes. And for this extended vortex model, we always have to keep in mind where we are in the species range, the history of the species, and the core spiraling of ecological interactions. And with this, I would like to end. I would like to, here is a picture of my lab group, people contributing with data to this talk, and the Swiss National Science Foundation as an important funding agency. And thank you very much. Thank you.
We have some microphones coming.
Oh, sorry. It's early. Thank you. That was a really fascinating talk. So I'm just curious to push your, the thought you had at the end about interactions and incorporating interactions into vortex models, for example. So we know that the more and more we learn, we realize these ecological interactions are way more complicated, and the networks, most of them are hidden from us. So how do you think one could do? It's important to do it, and yet so much remains unknown. So how do you think, what might be a good approach to start doing it, but cognizant of the fact that we are really unaware of many interactions?
You catch me at a weak spot, because I came to this conclusion while writing this talk, but it's not that I thought in depth about it. But I can tell you about for plants, especially these outcrossing plants, generally they're the ones that are going extinct locally. For example, across the European landscape. And yes, obviously they have a problem, probably because they have a self-incompatibility system that doesn't only preclude self-pollination, it also precludes mating with close relatives. And maybe that is a bit, made sense over evolutionary times. Nowadays it doesn't make sense anymore. It brings down population performance. But what is the other thing that we've just, maybe with these publications that came out, that we have this decline in terrestrial insects, in pollinators, and these are relatively recent studies, we start thinking, oh, maybe it has something to do with pollinators as well. So yes, here for plants, outcrossing plants, I think it's a worthwhile exercise to look whether, to what extent the declining abundance of pollinators contributes, for example, to lower seed set. So I talk now for my system to stay within my comfort zone. But maybe you have other ideas how this could be included.
Awesome talk. A question for you. Do we need to measure anything other than pi based on the correlations that you have? Can we can we sum it up to pi sort of explaining all of these metrics we're interested
No,
in?
I think after listening to your talk, I would say, of course, you need to take the it doesn't work for us. So what I showed you is obviously I picked the tightest correlations, the best figures out. So we have other estimates, other figures. So for us it works. But I think it is very important when we go back and look what explains pi most, especially once we started looking over the species range, it's this expansion or rearrange isolation. So something that happened a while ago, and probably if we think talk of Christina, so probably pi would be more going a bit deeper in time, and maybe we just pick up with pi that signal. It's the right time that we target with pi, I think. And I really think that the first point that was a bit, citing a study from 2006, maybe it's a bit really outdated, but I took this because I think it's really something that we need to work on, and we can only do it by bringing together results of different systems, different studies to generalize. So obviously, often the conservation effort is in, we have a particular species, it's in decline, and it's totally sufficient to have that data that is then very accurate because it's of that species. But obviously we have millions of species. We cannot do a population genetics for all of them. But if we want to bring it into conservation more regularly, probably we cannot do it by estimating all these things, load and the lacking signature of selection, etc. But if we knew what the right estimate of diversity was, to say, given this and in this particular species, probably with the correlation coefficients for which we in the future will hopefully have better estimates, because for more study organisms, with this percentage we can predict we have serious problems. That would bring us a big step forward, I think. So I think to answer it maybe more in a nutshell, the question, I think we need more data, and but then we would find out for what organisms and maybe what time we refer to when drift occurred is the right estimate, I think, but maybe there are other opinions.
Yeah, just to comment on that, I mean, pi is actually a historical estimator. It estimates things over about 4n coalescent generations. And so I think it tells you what happened in the past. It's the ghost evolution past. And I think for certain things that may have very long sort of lifespans and so forth, what's happening right now, what happens in the future, which is actually what I think is more useful for conservation biology, there can be a real disconnection. So pi, the story with pi is true, but it may be irrelevant. And so what you've done by actually demonstrating this functional sort of component of it is really important and really innovative. And I thought that was, a fascinating part of your talk, because you've answered the question by actually showing that it does matter. And I think that's really, really important to do and very hard to do.
Thank you.
I'm going to sneak in a question. So the correlations that you show are really amazing and how strong they are. I'm wondering if you've thought about the possible confounding factor that by studying like these range edge populations, that they're populations out of equilibrium. So should we expect this same sort of pattern in a sort of constantly small population that's in some population equilibrium? Or could that be a different scenario from the range expansion? So like I'm thinking about, you had those couple of ancestral populations that are not part of the range expansion. Did they show these same patterns as like small isolated populations or not?
Yes, so I obviously I cannot make any I would say probably most populations in nature are not at equilibrium, but so is it? Yeah, obviously what contributes, or where we see this low pies is in range edge populations, especially those that are far from this course of where the species seem to, from which it seemed to have recolonized. Yeah, so that carries it a bit, but not only. I think are the patterns a bit different at different edges? One thing that was actually interesting is this signature of directional selection in this old rear edge populations as compared to new edge populations. Those actually have a stronger signal of positive selection. So that is in there. It was sort of a very different pattern. Otherwise, they follow the general pattern. Yeah, but again, so our estimate of selection, I would love to think about doing it differently, doing it better. I don't think it's a bad estimate, but it would be nice to have different approaches.
Thank you for your talk. So I have just a question about the interpretation of alpha, and to me it makes sense, the interpretation based in the context of all the other results, which are super cool. But when I think of a classic McDonald-Kreitman test, I think of like you also want to compare to the fixed non-synonymous to synonymous ratio, which would allow you to kind of distinguish between selection and other types of forces. So I'm wondering, how would you interpret, like, for example, if you knew nothing else about this population, if you got a high level of alpha, how would you distinguish between positive selection and either reduced selection or like reduced efficacy of selection, relaxed selection? Like especially on the leading edge, right? If you have the leading edge of an expanded population, you have a higher degree of non-synonymous derived mutations. Mm-hmm. How do you distinguish between positive selection versus reduced efficacy of selection?
So I think what we look at is just the signature that remains. So we don't estimate, this is not estimating selection coefficients or the distribution of selection coefficients. This is just assuming selection may have happened or not, but what is the signature of positive selection? So it's always what we see as an imprint, and if we see, oh, that differs between populations, and in this case it differs depending on the history of drift, we assume that this lower signature is due to drift having a post directional selection. So it's based on assumptions. The estimate itself, it has its internal references. So the ratio of polymorphic, and then also two synonymous substitutions, polymorphic sites. Yeah. We did this also on the level of genes, the fraction of genes that show, so the same sort of method, but on the level of genes, and we find more or fewer genes showing a signature of adaptation in populations with a low pi.
Thank you so much.
Maybe one more.
I love your talk. I interested on the result that there increased mutational load in the edges, and then there is a reduced adaptation in the edges. But if I'm not mistaken, the effect for reduced adaptation is not as strong as the increased mutational load. Would that mean mutational load does not necessarily implicate reduced adaptation, or is just a different thing? So because like if you have too much deleterious mutation, I would naively think they would have really strong reduced adaptation, but apparently the effect is not that strong compared to the mutational load. Can we somehow connect that in this context?
Mm-hmm. So I think what is very important is to keep these two distinct hypotheses: reduced efficacy of purifying selection and reduced efficacy of directional positive selection. So, and it's basically the testing goes exactly for one or the other, and therefore I think insight on one doesn't necessarily tell us give us insights on the other. But I think the difference is so there is also if you think about your study organism and you think what it is adapted to, probably selection leading to adaptation, even local adaptation, very local adaptation, can be so strong that is stronger than drift that seems that tries to sort of erase the outcome of selection. So I think we have done a lot of phenotypic analysis on these populations, and we know, for example, northern populations at range edges as compared to very southern ones, they grow to larger size, they flower earlier because they have a shorter reproductive season. The ones from the very south, including the southeast, they're super well adapted to hotter, drier conditions. But these are probably there's selection in nature that is so strong as compared to drift. Drift is then not important, and therefore we still see, evidence for adaptation even in these small populations.
Okay, so it's safer to, keep the two hypotheses just different. I was trying to connect because it looks interconnected, but
it looks interconnected, but I think it is really something that we have to very clearly keep apart, I think. And with these estimates that we use, we do really try to focus on one or the other, but not on both.
Okay, thank you.
All right, thank you. Let's give Dr. Yvonne